Hi, everybody. I wanted to go ahead and talk to y'all a little bit and specifically talk to you about the citric acid cycle and move on to then a discussion of um, glycogen. Now, with that in mind, I want to go back to that. Yeah, I want to go ahead and jump in right here. Just a second. All right, so when it comes to the citric acid cycle, these are the things that I want you to know. Want you to know the enzymes that are involved, the cofactors that are involved, the structures of the different molecules, and where each of the carbons come from. Like I said in class the other day, one of the more important components of this discussion is I'm really interested in y'all knowing about the pyruvate dehydrogenase complex. Now, that movement of our three carbon molecule pyruvate ultimately to forming an acetyl-CoA molecule, and that acetyl-CoA can go in so many different directions, but here we're choosing to send it through the citric acid cycle. Now, the intent of that citric acid cycle, or the kind of goal or benefit of the citric acid cycle, is we're generating a bunch of this stuff: NADH, FADH2, and some more NADH. Now, keep of course keep this in the context of this is what happens with one pyruvate molecule. And if you're talking about glycolysis, well, you have two pyruvate molecules that are entering into the citric acid cycle. Now, this last item, where each carbon comes from, I touched on this a little bit, but I'd like to go ahead and elaborate upon that. If we look at the top here, our pyruvate molecule, the first thing that we're going to do with the E1 subunit complex of the pyruvate dehydrogenase complex, we have CO2. So we are decarboxylating pyruvate. And that uh, carbon dioxide that we're losing is this COO minus, this carboxylate ion. So the remainder, these two carbons, which remain from our pyruvate molecule, those are going to be what end up right here on our acetyl-CoA molecule. So if you look at that, this carbon, which was carbon number 2 in our pyruvate molecule, is this carbon right here, which is denoted by this double dagger symbol. That double dagger signal is shown here, 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 and then both here and here, because we are confronted with a molecule that is symmetrical once we get to succinate. So I want you to kind of keep that in mind, and then, basically be able to answer the question of, well, as time goes on and as rounds of the citric acid cycle are completed, where does that labeled carbon end up? So when you look at that labeled carbon, um, it's important to kind of visualize where that is. Now, if we jump in at malate, we've got a label here, we've got a label here. Well, this carbon, which I'm going to label A, just so we have a little sense of organization here, well, that is that COO minus. Now, where does that end up? That ends up right here. That remains right here. So this is basically running through the citric acid cycle for a second time. And that label, well, that label is going to be lost at step number 3. So effectively, if you have a label on a carbon in this position, this carbon number 2 in this pyruvate molecule, that label, whenever I talk about a label, I mean like a radioactive label. So a C13 label or something like that, an isotopically labeled carbon atom. Well, that's going to be removed from our system when we decarboxylate at isocitrate dehydrogenase. Whenever the carbon has made it through this cycle one full time. So then you can ask the question, well, is there still a label? Is there still a radioactive label on this alpha ketoglutarate molecule? There absolutely is. Because if you remember, going back to this malate, before we complete one whole cycle, we've got a label on this carbon that I have labeled A, and we also have one at this carbon, which I'm going to choose to label B. So it's important to kind of track those, see where those labels are, where they're going, and everything like that. But these are the major components and players of the citric acid cycle: the enzymes and the cofactors, the structures of the individual molecules, and where each one of those carbons come from. Now, only 3 of the enzymes of the citric acid cycle are completely irreversible. Those enzymes are citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase, which, if we go backwards and look at those, we have citrate synthase, our first step. And let me go ahead and clear all my other markings up here. Okay, so this one, irreversible. You see that with the equilibrium arrow. Isocitrate dehydrogenase, irreversible. And alpha ketoglutarate dehydrogenase, also irreversible. So we have a total of 3 steps, 1, 3, and 4. What's happening at each one of those steps? I think that's an important question to ask yourself. Well, 1, we're forming our big 6-carbon or our 6-carbon molecule citrate, and we're releasing CoAsh, or the reduced form of our coenzyme A. Next up, isocitrate dehydrogenase, we have our decarboxylation and the formation of NADH. Then alpha ketoglutarate dehydrogenase, well, we also have a decarboxylation and formation of NADH. So if you wanted to think about this as what are good ways to kind of imagine or remember which steps are going to be irreversible, think if there's a decarboxylation and the formation of NADH, both of those things happening, then there's pretty good reason to believe that we have an irreversible step. Because there are other steps where NADH is generating, but it's not NADH and CO2. All right, so those are our 3 steps. I want you to know those 3 steps are irreversible steps. When it comes to thinking about which enzymes and which structures should I focus on, I would focus on those irreversible steps. What is the substrate and what's being formed? Add to that, are there any unique cofactors at any of those steps? I think that's a good place to kind of focus your studying, as opposed to saying, I've got to learn every single structure in this pathway. I would focus on those irreversible steps because those are kind of the interesting ones. Now, if you look at what we have with respect to our delta G naught prime, or our free energy of those reactions, we have negative 31.5, negative 21, negative 32, or 33. The magnitude of those steps is they are so favorable in the forward direction that those delta G naught primes are considerably higher than the others. If you compare that to something like succinyl-CoA synthetase or fumarase, those are both favorable reactions, but not nearly as favorable as those other steps that we looked at. Now, an important part of any metabolic pathway, and in particular the citric acid cycle, is understanding the regulation of those steps. Just a second. So, of all these different steps, one thing that's worth noting and one thing that's worth considering is, what's the point of these metabolic pathways that we've talked about so far? Largely, our focus has been in the formation of ATP. We want to generate ATP. We want to take glucose and generate ATP. And you could kind of think about this as the need for doing these pathways is to generate ATP. So if you have no need for ATP, is there a need to proceed with that pathway? And the answer, in some respects, is kind of no. So NADH is a molecule that we can use ultimately to generate to generate ATP in the electron transport chain. Now, NADH is a regulator of a number of different enzymes. For instance, NADH negatively regulates the pyruvate dehydrogenase complex. So it prevents the transition from pyruvate to acetyl-CoA. So NADH is going to interrupt the formation of acetyl-CoA. What else is NADH going to do? It's going to inhibit, at this first point right here, it's going to inhibit our first step in this pathway where we're taking acetyl-CoA and oxaloacetate and making citrates or citrate. So our citrate synthase. This is going to be NADH is going to inhibit that step. Okay. What else is NADH going to do? NADH is also going to inhibit the step where we're generating alpha-ketoglutarate, which would be our isocitrate dehydrogenase. So NADH is also going to inhibit this step right here. After that, our step that leads to the formation of succinyl-CoA, which is our alpha-ketoglutarate dehydrogenase. So NADH is going to be inhibiting citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. And you can kind of think about this as, well, we have three enzymes that are irreversible. Those are very favorable reactions in this pathway, and they are very negative, or they are negatively impacted, and they are negatively regulated by NADH. So the molecule that is kind of the purpose for this pathway is a very tight regulator of this pathway. Now, that's what this model is showing. In addition to that, what we have is we have other molecules that are going to feed into that regulation. For instance, succinyl-CoA and citrate will also inhibit citrate synthase, or CS. So I'm going to go ahead and label this as CS. The citrate synthase is a point of regulation. It's regulated by NADH, citrate, and succinyl-CoA. So that succinyl-CoA is almost like a downstream regulator that if citrate doesn't regulate it enough, then succinyl-CoA will come in and kind of pump the brakes on it. Succinyl-CoA is going to also serve as a regulator of our alpha-ketoglutarate dehydrogenase, labeled right here, alpha-KGDH. And then ATP is also going to be a regulator. ATP is going to be a regulator of our isocitrate dehydrogenase. So, DH. So when you look at this pathway, one of the things that I want you to think about is, I want you to think about this pathway right here, or this list of our enzymes. The ones that stand out, or the ones that I think should stand out to you: citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. Those are three irreversible steps. Those are akin to our hexokinase and phosphofructokinase. Those are steps that are very favorably going in one direction. Well, those are also junctions of regulation. Now, we're talking about TCA cycle regulation. We haven't gotten into glycolysis and gluconeogenesis regulation because that's a little bit more complicated. But this arguably is kind of straightforward. These three enzymes are very tightly regulated. And they are tightly regulated by products of this pathway. In terms of, in terms of molecules that activate this pathway, we have calcium. Calcium is a key activator for the conversion of pyruvate to acetyl-CoA. Calcium is also an activator of isocitrate dehydrogenase. Calcium is a regulator or a positive modulator of alpha-ketoglutarate dehydrogenase. So our targets for kind of negative or depression of activity of the citric acid cycle, we're targeting those irreversible steps. In terms of activation of this pathway, we're targeting 2/3 of those key regulatory steps. So this regulation is, if a product is present, it's going to inhibit the reaction from taking place. It's going to inhibit that pathway from moving forward. So that's what I want you to take away from this. I really want you to look at this cycle right here and line it with this list of your enzymes, because I think that that will be helpful to think about which are the enzymes that are arguably the most important ones in this pathway. Now, pyruvate dehydrogenase, in addition to being regulated by the presence of NADH, pyruvate dehydrogenase is a molecule that, in order to be activated, has to be dephosphorylated. So whenever you're adding OPO3 to a molecule, that is referred to as phosphorylation. Now, there are enzymes that do this, that add phosphate groups. Those enzymes are known as kinases. There are protein kinases. There are also sugar kinases. So pyruvate dehydrogenase is going to be phosphorylated or dephosphorylated. Now, if you look at this model, we're seeing this E1. This is in reference to that E1 subunit. When that E1 subunit has received a phosphate group from ATP provided by pyruvate dehydrogenase kinase, well, then E1 cannot catalyze its reaction. So what that means is that this enzyme right here will not decarboxylate pyruvate. Okay. So E1, when phosphorylated, will not decarboxylate pyruvate. There's another enzyme, pyruvate dehydrogenase phosphatase, which hydrolyzes that phosphate away. So E1 is not phosphorylated, and therefore it is active and it can phosphorylate it can decarboxylate. Ugh. So we have E1, which is regulated by a couple of different things. One, E1 is regulated by the presence of NADH. So when when the pyruvate dehydrogenase complex senses NADH, then it stops its activity. Okay. Likewise, E1 of the pyruvate dehydrogenase complex, whenever it gets phosphorylated, it will stop its activity. So what we have here is a multi-pronged approach to the regulation of the pyruvate dehydrogenase complex. Now, pyruvate dehydrogenase kinase is an example of a protein kinase, because what that molecule or what that enzyme is doing is it's grabbing ATP and transferring a phosphate group onto another protein. A sugar kinase, by contrast, is an enzyme that transfers a phosphate group from ATP onto a sugar, like what we've previously seen with hexokinase and phosphofructokinase. Those are both examples of sugar kinases. All right, so this adds to the regulation of our citric acid cycle. The pyruvate dehydrogenase complex can be regulated by NADH, which is a product of that pathway, or this other enzyme, pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase. So thinking about when are circumstances or when are instances that you want to generate a lot of NADH. So if you have a very low concentration of NADH, that's an environment where you're going to want to generate, you're going to want to have an active pyruvate dehydrogenase complex. So you're more than likely going to have the dephosphorylated E1. So these kind of work in concert with one another. So if I said E1 is phosphorylated, then chances are, so when E1 is phosphorylated, then you have, or you more than likely have a very low concentration of NADH. Now, it's possible that you have some NADH there, and you have an active form of the pyruvate dehydrogenase complex. You have this phosphatase that's active, but those two things are working. They can work in concert, but they also can work. They're kind of they're let's see. They're both working on the same thing. They're both working on the same enzyme and enzyme complex, but they're not forced and driven by the same thing. So the phosphatase and the kinase are activated by an entirely different signaling cascade or entirely different stimuli than, say, the presence of NADH. Okay. Now, in a nutshell, the pyruvate dehydrogenase complex, whenever acetyl-CoA and NADH are present, chances are you're going to have a diminished activity of the pyruvate dehydrogenase complex. Whenever you have calcium, insulin, and ADP present, well, then your pyruvate dehydrogenase complex is higher because what you're trying to do is extract as much energy from glucose as possible. All right, so we've kind of stepped our toes gently into the pool of regulation of the metabolic pathway. It's very complicated. We've looked at a couple of or a fairly narrow view of or a narrow scope. We've had a narrow scope of our enzymes and metabolic pathways. But we added another wrinkle whenever we introduce these kinases and phosphatases, because they could be activated or inhibited by something like NADH, but that doesn't mean that they are acting in the same way on the pyruvate dehydrogenase complex. Okay. So with that said, I want to kind of transition to take a little bit more of a global perspective on what we've been talking about. And by doing, or the way that I'd like to do that is by talking about glycogen. Now, glycogen is our primary sugar storage system in the body. In the muscle, 1 to 2% of the weight is glycogen, and it can be pretty actively broken down so that you can get glucose from that. In the liver, on the other hand, up to 10% of the weight is glycogen. And the reason that you have a lot of glycogen there is in order to help the liver maintain approximately 5 millimolar blood glucose concentration, which, just to put into perspective, that's millimolar. I always like to think about, in Gen Chem 1, everything that you talk about is in like a molarity scale. You're talking about a 10 molar solution or a 50 molar solution. Here we're talking about 0.005 molar solution, so a fairly low concentration. The liver has approximately a 12-hour supply of glucose, and that can be supplemented with gluconeogenesis when needed. So amino acids can ultimately be shuttled for the formation of glucose. Fatty acids, if you have virtually no glucose, can ultimately, because with fatty acids, you can get to acetyl-CoA. Acetyl-CoA can then be used to make pyruvate. Pyruvate can then go back up to make glucose through gluconeogenesis. Now, glycogen is a very key and important energy storage system in the brain. You are able to use that glycogen fairly easily, changing your right now my brain's not working. Sorry. It can be rapidly mobilized when O2 is limited in the brain. So when you think about, or when we've talked about glucose breakdown, we don't require O2. And so whenever the O2 availability is very low, well, then you can take something like glycogen and break off those individual glucose molecules so that you can kind of keep the system moving in an oxygen depleted environment. So the structure of glycogen is what I'd like to focus on and what I want y'all to focus on. Whenever we talk about a glycogen molecule, and actually whenever you talk about any sort of dye or polysaccharide, there's two major ends to it. There's a reducing end and a non-reducing end. One thing that I'd like you to look at here is this reducing end. What we have here is known as a hemiacetal. So that hemiacetal, what that can do is that ring, that glucose ring that I have a dot in the middle of, that can open and close. You have that interconversion between the alpha and the beta configuration of that glucose. And that interconversion is known as your immunorotation. The contrary, on the opposite end of this molecule, we have these kind of end of the chain glucose molecules. These are referred to, or they have what is known as a non-reducing end. And everything in between, well, it can't open and it can't close or anything like that. These non-reducing ends, well, you can add on to them, but their level of reactivity is fairly low. Now, what this glycogen molecule is displaying is a couple of different things. One, like I've alluded to, the reducing and non-reducing ends, but then we've also got these alpha 1,4 linkages. The 1 is coming from here. The alpha configuration is because this is pointed downwards. This is carbon number 4 of a glucose molecule. It is also pointed downwards. So we have this alpha 1,4 linkage. This is a glycosidic linkage. We have 1, 2, 3, 4, 5, 6 alpha 1,4 glycosidic linkages. We also have an alpha 1,6 glycosidic linkage. Now, this alpha 1,6 glycosidic linkage is a branch point. So this is where our glycogen molecules get much more complicated. Now, this is different from something like cellulose, because cellulose is going to have beta linkages, which that's one thing that's going to make them different. But cellulose is also a completely linear, non-branched sugar molecule. Glycogen, on the other hand, you're going to have a couple of branch points, and those branches can have branches, and those branches can have branches that have branches. Glycogen structure, glycogen molecule, is ultimately going to look very large. It can be very complicated. The hallmark of a glycogen structure or glycogen molecule is you're going to have one reducing end. I circled those reducing ends on the bottom of these two kind of macromolecular models. And that's just the freely accessible carbon number 1. Now, of these two models, we've got the one on the left here, which I'm going to label L. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. 16 non-reducing ends. So 1 reducing end, 16 non-reducing ends. The number of non-reducing ends is really all related to the number of branch points that the molecule has. These branch points are important and significant because these branches have different bonds, and those different bonds are processed in different ways. Those bonds are going to be broken in different ways. For instance, we have an enzyme known as glycogen phosphorylase. What glycogen phosphorylase is going to do is it's going to take the straight chain of a glycogen molecule, and it's going to cleave off glucose after glucose after glucose. And it does so with the help of inorganic phosphate. And it's going to release a molecule known as glucose 1-phosphate, which, when you think about glycogen as your storage molecule and breaking that glycogen down, and you end up with glucose 1-phosphate, how does that relate to glycolysis? Well, glucose 1-phosphate, we don't actually use that in glycolysis. We use glucose 6-phosphate. We use fructose 1,6-bisphosphate, but we don't use glucose 1-phosphate. So that molecule is going to be processed before it can be utilized for glycolysis. Now, glycogen phosphorylase's activity is irreversible. What it is looking for, it is looking for a straight chain, and what it's going to do is going to remove one glucose at a time from a non-reducing end. And that is going to look for, or it's going to be able to kind of cleave off glucose molecule after glucose molecule until it reaches a point where it's within basically 5 glucose subunits of a branch point. So it's not going to be able to, basically, you could think about the active site as soon as it gets close to that branch point, it has to stop because it cannot manipulate around that branched molecule. So effectively what that means is if you have a linear molecule, glycogen phosphorylase can do the job. But as soon as you introduce a branch, then you have to manage that branch in a certain way. Inorganic phosphate, like I said, or PI, is used as opposed to ATP. So we don't have to use any ATP for this hydrolysis. What this is showing here is our mechanism for glycogen phosphorylase. We've got a inorganic phosphate molecule right here and our glycogen molecule. Then we have through an acid-base catalyzed, or actually this would be a covalent mechanism. What we're going to do is cleave off that individual glucose molecule, and with the help of this cofactor known as pyridoxal 5-phosphate, or PLP, that's going to help us cleave off those individual glucose molecules and process them as they proceed, as we need to. This is going to help us simply use inorganic phosphate, or PLP is going to help us use that inorganic phosphate to cleave that off. If you look over here on the left-hand side, this model here, and here, what we have is this PL, which is where our PLP molecule is going to be anchored within the active site of our enzyme. And so that is basically going to put this phosphate group out so that it can be attracted to this inorganic phosphate molecule that we can then use to remove those individual glucose molecules, or those individual glucose subunits. Now, glycogen phosphorylase, our enzyme that cleaves off the individual glucose 1-phosphate molecules, is going to be very tightly regulated. So if you think about it as glycogen is a storage molecule, then you don't, it's almost like it's a, it's almost like a savings account where it's your rainy day fund. You don't want to go into that unless you really need to. Glycogen phosphorylase is going to be regulated by a couple of different mechanisms. One, covalent modification. So this is an enzyme that is regulated by phosphorylation. So phosphorylation is one method of covalent modification. Others, just to put into context, but I'm not going to ask about these, would be methylation or acetylation. Adding small functional groups to the molecule is going to change how it acts. So what this is showing on the right-hand side, I'm going to put a box around this, is let's go down here. I'm going to put a star by our enzyme that we're focused on, glycogen phosphorylase. Glycogen phosphorylase, shown on the right-hand side in green, when phosphorylated, is more active. Okay, when dephosphorylated, that glycogen phosphorylase enzyme exists as glycogen phosphorylase B, the inactive form of it. Okay, so what that means is that glycogen phosphorylase has to be phosphorylated in order to be active. Okay, so anything that removes that phosphate is going to diminish its activity. Anything that phosphorylates it is going to activate it. The enzyme that does that is an enzyme known as phosphorylase kinase, sometimes referred to as PK. Phosphorylase kinase is a protein kinase because it phosphorylates glycogen phosphorylase. So phosphorylase kinase activates glycogen phosphorylase. The funny thing is, and the complex thing about protein kinases is phosphorylase kinase itself, being a kinase or a protein kinase, is also the target of another protein kinase. This enzyme upstream here, protein kinase A, PKA. Protein kinase A is a master regulator, and it is going to phosphorylate phosphorylase kinase in the right environment. So it will phosphorylate phosphorylase kinase, which phosphorylates glycogen phosphorylase, which leads to the activation of breakdown of glycogen. So think about this as when protein kinase A or PKA is activated, that is going to activate phosphorylase kinase. When phosphorylase kinase has been activated, then glycogen phosphorylase is going to be activated. So this is basically a nice little light switch of, as soon as protein kinase A is activated, then there's a sequence of events that lead to glycogen breakdown. Anything that leads to the opposite of any of those things is going to lead to preservation of glycogen. Okay, so there's one of the ways in which glycogen phosphorylase is going to be regulated. Now, glycogen phosphorylase is also an allosterically regulated enzyme. So it is going to transition between the T state and the R state. If you remember allostery or allosterically regulated enzymes from a previous discussion of hemoglobin, not from this class, but your first biochemistry class, hemoglobin is an allosteric protein. T state, the R state. The T state is your inactive state, it's the tense state, and the R state is the relaxed or the active state. Deoxyhemoglobin, for instance, is the deoxygenated hemoglobin. That's the inactive form of hemoglobin. That's your T state. Oxyhemoglobin, your active form, is your R form. Now, AMP, adenosine monophosphate, is a positive modulator of glycogen phosphorylase. AMP is going to push the equilibrium towards the R state of glycogen phosphorylase. Glucose 6-phosphate, on the other hand, is a negative regulator. It is a heterotropic effector that is going to lead to the formation of the T state of glycogen phosphorylase. So T state and the R state of glycogen phosphorylase. If we're talking about it being the R state, well, then you can go ahead and expect glucose 1-phosphate being generated, glycogen chains being broken down. Glucose 6-phosphate, on the other hand, well, that is one of our intermediates in glycolysis. And if you're doing glycolysis, if you're going through glycolysis, do you think that you need to be breaking down, or do you think that you need to be breaking down your storage molecule, your glycogen, in order to, or sorry, do you think that if you're going through glycolysis, that means you have an adequate, or you have sugar that you're trying to break down, free glucose that you're trying to break down. Would it make sense to go to your reserves? It's almost like you've been given $100. When you have that $100 and you're going to spend it on something, do you need to go back to the bank and get more money? You don't, if all you're going to spend is $100. Of course, that's a complicated question. Anyway, some of the ways in which glycogen phosphorylase is regulated. Covalent and allosteric regulation. Our next enzyme, the debranching enzyme. We talked about glycogen phosphorylase as our enzyme that removes glucose molecules one at a time in the form of glucose 1-phosphate. But then we have this branch point. Well, the debranching enzyme, cleverly named, is the enzyme that tends to that. The debranching enzyme, what it is going to do is it's going to see these branch points. And as soon as that branch point, or as soon as that branch is 4 subunits long, it's going to cut 3 of them off and move them to the main chain. Whenever those are removed to that main chain, then what we're going to be able to do is glycogen phosphorylase could then again begin working on this molecule. But the debranching enzyme, what it's also going to do is it's going to remove this final branch point. So think about the debranching enzyme looking for a chain that's 4 units long, or a branch point that's 4 units long. 3 of them get cut off and move to the main chain. The remaining branch point with that alpha 1,6 linkage, that's going to be cut off and release this glucose. So this is one of the key differences between glycogen phosphorylase and the debranching enzyme. Glycogen phosphorylase makes glucose 1-phosphate. The debranching enzyme, on the other hand, makes glucose. What this leads to is when you think about the work done on a glycogen molecule, what you have ultimately, or if you break down a glycogen molecule completely, 90% of the glucose in that molecule is going to be left behind as glucose 1-phosphate. The remaining 10% is going to be broken down into glucose. So another way that you can look at that is 90% of the molecule will end up or is going to be subject to glycogen phosphorylase, whereas 10% of it is going to be kind of dealt with by the debranching enzyme. Okay, now deal with our last component here, that glucose 1-phosphate. The glucose, which we haven't talked about, galactose metabolism yet. The glucose 1-phosphate can be isomerized and converted from glucose 1-phosphate to glucose 6-phosphate. In order to do that, what's going to happen is this enzyme phosphoglucomutase is going to phosphorylate glucose 6, or at the 6th position, leading to this intermediate that's glucose 1,6-bisphosphate. Then the same enzyme, this phosphoglucomutase, is going to remove the phosphate from carbon number 1, and we're going to leave behind glucose 6-phosphate. So what we have here is this intermediate that's kind of funny, glucose 1,6-bisphosphate. It's a doubly phosphorylated 6-carbon sugar. Have we seen a doubly phosphorylated 6-carbon sugar before? We absolutely have. F16BP. Nope. Fructose 1,6-bisphosphate. Okay. Is fructose 1,6-bisphosphate the same as glucose 1,6-bisphosphate? No, it's not. Fructose 1,6-bisphosphate is a ketose, a doubly phosphorylated ketose, whereas glucose 1,6-bisphosphate is a doubly phosphorylated aldose. So these sugars are different. They're both 6 carbons. They're both doubly phosphorylated. But is glucose 1,6-bisphosphate a good substrate to be used by our aldolase to break it into our two molecules of glyceraldehyde 3-phosphate, or one of that and then one of DHAP? No, it's not. So unfortunately, we can't really intercept that glucose 1,6-bisphosphate and plug it into glycolysis. But phosphoglucomutase is what takes our glucose 1-phosphate to make glucose 6-phosphate. So essentially what we're looking at when it comes to glycogen breakdown, basically 3 enzymes: glycogen phosphorylase, the debranching enzyme, and phosphoglucomutase. If you have those 3 enzymes working, the breakdown of glycogen is fairly simple and straightforward. You break off individual components, making glucose 1-phosphate, until you reach a branch point. That branch point is then dealt with by the debranching enzyme, and the product of this subsequent breakdown of this glycogen molecule is dealt with by phosphoglucomutase. So there's one enzyme in the liver, glucose 6-phosphatase. Now, glucose 6-phosphatase is an enzyme found in the liver almost exclusively. And what it is going to do is it's going to take glucose 6-phosphate to produce glucose and inorganic phosphate. Now, this is an irreversible step. This irreversible step allows excess glucose from glycogen breakdown or gluconeogenesis to enter the bloodstream to ultimately be transported to the brain and other critical tissues. So this enzyme being pretty exclusive to the liver is another way of bringing back the importance of the liver for maintenance of your blood glucose levels. So this is not something that's in every other tissue that breaks that. It's not in muscle tissue. Muscle tissue has a little bit of glycogen, so it's going to break those down. And it's going to lead to formation of glucose 6-phosphate because that muscle tissue is going to take that glycogen, break it down. For instance, if this Sunday you're running the Austin Marathon or half marathon, well then your muscle tissues are going to be using this phosphoglucomutase. Muscle tissue is not going to be using this glucose 6-phosphatase because your muscle tissues want to use that up very quickly. And by getting glucose 6-phosphate, they can plug that straight into that they can go ahead and convert that glucose 6-phosphate to fructose 6-phosphate. We're not really concerned with that so much with your muscle tissues and glucose 6-phosphatase. All right, well, I hope that was helpful. And if you have any questions about the regulation of the citric acid cycle or glycogen metabolism, please let me know. Have a good one.